Welcome back. In the last few lectures, we have discussed how different DNA tools have revolutionized the field of biotechnology and genetic engineering. Many times I have given you examples which are especially on the medical field. Today we are going to really focus an area based on the plant biotechnology and how genetic engineering has contributed towards that. If you go back and think about my very first lecture, where I started emphasizing the need for various type of global challenges, think about abiotic stresses. For example, salinity, heat, drought, all of these are abiotic stresses which are affecting our land, the cultivation area severely throughout the world. Almost 20% of agricultural land is getting affected because of the salt stresses, and large volume of the overall earth is getting desertified because of different drought situation which is happening throughout the world. As you can see here in couple of images, various type of natural calamities like tsunami, Katrina, many of these natural catastrophe, they leave huge impact not only on the climate that time, but also for the years to come. The whole land becomes barren and nothing could be grown on those kind of field. So, these abiotic stresses, they impose major limitations on food production in whole world. If you think about human population which is projected to grow almost 50% by 2050, and the land which is being used for the food crop cultivation is getting reduced over the period, and it is also getting subjected to various type of environmental stress conditions. So, how are we going to address the environmental impact on food production? So, we are facing many challenges globally. The world food grain production must be doubled by year 2050, because we have to meet the demand for the growing global world population, and we have to increase the food production on the limited land area which is available to us. And on one hand, the water resources are getting diminished, a lot of lands are getting affected because of the salinity, drought, cold and many stresses, and on the same small area which is available for the food crop production, we have to increase the crop productivity. So, what are the biotechnological solutions available to address this challenge? For example, we can increase the crop yield, we can reduce the need for the chemical and water inputs. We have to also increase the resistance for the plants towards the abiotic stresses such as salinity and drought. Also, the plant should be more tolerant for the various type of insects and pests. So, in order to improve the crop traits by using biotechnology, there are a couple of case studies I am going to discuss with you. The genetic modified organism or the GMOs is one of the common and a very hot topic which I am sure you often read in the newspapers, which you often hear about it. This is one of the GMO is one that has acquired one or more genes from another species, and this can be done by using the genetic engineering artificial means, right. So, in some way that we can actually cross the barriers of different species, and we can move genes from one to other organism for different type of traits, and that is what is being utilized and exploited in the field of biotechnology. So, let me take you the very first case study on developing the stress tolerant plants and especially the salt tolerant plant. So, as we discussed that there is a need to have the plants which could survive even if you have a hard salt condition, hard drought condition. Still, if those plants can live in this kind of environmental situation, then probably we can have the more crop yield. So, traditional breeding which is commonly employed has been unsuccessful majorly to generate the highly tolerant saline or the drought tolerant crops. So, can biotechnology and genetic engineering provide some solution to that? So, in order to do that, in this study the researchers looked into the pea plant, and the pea plants were subjected to different type of stress conditions from 0 to 75 and 150 millimolar of the salt condition, to look into the the medium and the high salt concentration, that how that affects the pea plant. And now the root and the shoots were excised, and as you can see the roots are getting quite affected in these as the salt is increased concentration. So, proteins were extracted from these roots samples, and now the proteins were separated using a gel format, which is 2-dimensional gel, where proteins are separated based on the isoelectric point and their molecular weight. Lot of spots which you can see on these gel images, which are actually showing you that the proteins obtained from the roots which are affected from the salt conditions. Now, what kind of protein changes are being seen as the salt concentration is increasing? After doing this analysis, as you can see in the highlighted circle part, that a group of protein looks quite highly elevated, and those protein definitely showed that increasing response to the salt stress condition. And of course, one will be curious to know that what those proteins are, right. So, another experiment was done where a salt sensitive and salt tolerant Arachis hypogea, another type of the plant was taken, and again their proteins were compared using proteomic kind of investigation. And as you can see on the left side, the protein obtained from these callus were separated on these gels, 2-dimensional gels, and again on the lower side a group of protein, a set of proteins are highly expressed, which is shown in the circle, red circle here, which is very similar to the kind of pattern which you have seen in the last slide, where I showed you the pea plants getting affected because of the high salt condition. So, looks like in the similar area of same molecular weight and same pI, a set of proteins are getting highly overexpressed, and what those proteins are. As we will go in the next module, I will talk to you about how protein technologies work, and that time you will be also exposed to the mass spectrometry based proteomic investigation. But for the time being, to keep the context here, you should know that the mass spectrometers are one of the powerful analytical tools which can be used to identify the proteins of interest, where you can look at the peptide sequences and do the database search to identify what are the proteins. So, in this case, let us say from this gel piece you have excised the protein sample, you have digested them to make peptides, and now those peptides are subjected to the mass spectrometers to identify what is this protein of interest, because this is what you want to identify, that what this protein is. So, when researchers did this work, they identified this protein is a group of family of the protein which is pathogen resistant 10 protein, PR10 protein, and there are a couple of members of those proteins which were identified. Now, can we use this information and use it back in the genetic engineering approach, is what is shown you in the next slide, where an agrobacteria mediated transformation system was used to do the genetic engineering approach. Now, this protein PR10, which is coded by a gene PR10, can we introduce that in the plant system, and whether this particular gene could boost up the plant's tolerance to the environmental stress condition, for example, salt stress condition. And there are different the gene family, for the PR10 is a large gene family. This specifically one member PR10.1 was used in this particular experiment. So, you have been we have been discussing about the vector map. Now, you can see that this vector map, different restriction sites are there, the promoter site is there, and now we want to introduce this PR10 gene of our interest. We are using here one of the agrobacterium mediated transformation system, in which the PR10 gene construct has been introduced now in the bacteria, which is now getting co-integrated this plasmid inside the plant cell. How this exactly work, I will elaborate in some more slides later on. But ideally, you are trying to introduce this particular gene of interest in the plant by using this transformation system, which is agrobacteria mediated transformation. Couple of images shown you here, that how some of these things are experimentally done in the lab, where when you want to introduce these genes from one system to the system, then you have to do the series of the experiments based on the tissue culture based methods in the lab inside the greenhouse condition, and then slowly you can take these small implants where your gene of interest has been introduced. Now, you are growing them further, and you are trying to provide them the limited environmental conditions so that they can grow in the beginning, and then slowly you want to test the effects of the gene which you wanted to test out. So, in this case, when researchers tested the utility of PR10 genes in a different crop, because you have identified the gene from pea, what is the impact of this particular gene tolerance into another plant species, which is Brassica napes or the canola plant. which is another plant of highly economical significance. So, if you now look at the seedlings of the non-transgenic and the PR10 transgenic, they show quite a bit of difference, that the no salt is of course both are surviving quite well, but as the salt concentration is increased to 75 millimolar, in this case you can see that the transgenic plant which has incorporated the PR10 gene of interest is having much more tolerance. This is another image which is showing you the greenhouse grown plants now. In the high salt condition, even up to 150 millimolar, these plants were able to grow and survive as compared to the non-transgenic or the control plants. And then similar utility was tested in the Arabidopsis plant, and now you can see that these Arabidopsis seedlings are quite able to tolerate the high salt condition. So, again this gene is doing something in the plant which is kind of increasing the plant tolerance towards the salt concentration, and that was seen in the case of canola, it is seen in the case of Arabidopsis. Let us come to another example of very successful example, which is golden rice. As we are all aware that the large number of children become almost blind because of the vitamin A deficiency, and can genetic engineering provide some solution to create the rice, and one of that attempt was done, which is known as golden rice. I have shown you here two highly cited publications of this area, where they have engineered the provitamin A or the beta carotene biosynthetic pathway into the carotenoid free rice endosperm, and another study which was which has shown that you can improve the nutritional value of golden rice by increasing the provitamin A contents, and they have expressed in this case a Cy transgene which increase the carotenoid contents of the maize callus. So, in these studies what they did, they wanted to look at the transgenic variety production, which could be supplemented with two daffodil genes, which can enable the production of the grain containing beta carotene, which is a precursor of vitamin A. The daffodil genes encode phytoene synthase or the Cy, and this golden rice too contains the Cy gene from maize and carotene desachuratase from Erwinia iridivora. And overall they observed an increase in the total carotenoid of up to 23 folds. So, after discussing the successful example of rice, which is golden rice, which is grown in the large areas and has really changed the nutritional quality of the rice. Now, let us come to the third most cited topic, resistant to insect pest. The large number of crops and the food crops in fact even get affected because of the pests and insects which affect in the when they are grown in the field. So, the pesticides and insecticides are used to protect the plants from the impact of these pests and insects, which are very harmful for the plants. However, these insecticides and these chemicals are quite detrimental for even human health. So, since 1940s, people have started reporting many drawbacks of the existing insecticides, including DDT, which lack a specificity. It is non-selective, it is actually toxic to the human health, as well as many of the non-target animals. Once you spray these type of chemicals on the plant, then they come from water inside the soil, and they also get bioaccumulated. You can see that they are getting persistent in the environment. It is very tough to remove them from the environmental conditions. And of course, there is a huge cost of spraying these chemicals on the plant to protect them from these pests and insects. So, can genetically engineered insecticide crops could be created, which can boost the immune system of the plant for that matter, so that the plants can now tolerate these kind of infections and still survive in the field. To do that, a highly successful example comes where people have used the gene from Bacillus thuringiensis or the Bt gene. So, Bt is a ubiquitous spore forming soil bacterium which produces insecticidal protein crystals, which is known as Bt toxin. This is one of the endotoxin crystal protein which is produced during the sporulation process of this bacteria. It was discovered in 1901 by scientist Ishiwata on silkworm. During 1928 and 31, they performed some experiments to control the corn borer, and this was one of the best natural gene product which was used to control the plant pest. and that gives them an idea that can this type of genes be used to protect the plants from the deleterious effects of the insects and pests. So, how a genetic engineering approach could be used here? So, there are three primary components of genetic packages which were inserted into the plant. The protein gene, which is the Bt gene, which is going to produce the cry protein, a promoter sequence which controls where and how much of the cry protein has to be produced in the plant, and a genetic marker which can identify the successful transformation experiment, for example, the antibiotic resistance, which can be used for the screening purpose. So, let us look at how the tumor inducing plasmid or TI plasmid could be used to produce transgenic plants. Let me explain you this in more detail in the following animation.
When a plant gets injured, there is release of the phenolic compound acetosyringone, which is detected by Agrobacterium tumefaciens. Upon detection, the virulence genes of tumor-inducing TI plasmid get expressed, which encode the enzymes that are essential for transfer of the tDNA into the nucleus of the plant cell. Once the tDNA gets integrated with the plant chromosome, there is release of cytokinins, auxins, etc., which brings about tumor formation in the plant. The useful property of infection by Agrobacterium has allowed several foreign genes of interest to be introduced into plant cells as per the requirement. One plasmid of the cell is a TI plasmid without the tDNA. The other plasmid contains the gene of interest along with antibiotic resistance genes placed in between two repeat units that are essential for gene transfer. The gene of interest, such as genes for pesticide resistance, better yield, etc., invades the plant at the site of injury. Once this happens, the foreign gene gets inserted into the plant DNA, which is confirmed by plating on to agar containing the suitable antibiotic. Only those which have taken up the gene will grow on such plates.
Agrobacterium tumefaciens mediated a transformation was used here. The TI plasmid was isolated from the bacteria. Now, the foreign gene of interest, which is Bt gene in this case, was inserted into the tDNA. The recombinant plasmids were introduced into the cultured plant cells by the process of electroporation. Only those plant cells which took this plasmid, where the tDNA was integrated into the cell's chromosomes, those were taken forward. So, what is the mode of action for this Bt gene? Initially, the protoxin parasporal crystals or delta endotoxins, they are ingested by the target insect. Now, these protoxins are activated in their gut because of the alkaline pH which is there, which activates this particular toxin. So, after these crystals have been taken by the insect, when it goes to their gut, now that environment of alkaline pH is activating this toxin which was inactive earlier, and now the cry protein is going to bind to the receptors in the mid gut of insect, which leads to the death of this particular insect. So, the cry proteins or endotoxin, they bind to the receptors in mid guts of insects, they form ion selective channels, causes epithelium cells to swell, and they lyse because of the influx of ions and waters and lead to the death of this insect. There have been large number of successful examples for transgenic Bt crops which have been produced worldwide. I have shown in this table here the transgenic Bt cotton, for example, Bt corn, Bt potato, Bt soybean, Bt tomato, all of these crops having different type of cry proteins which were inserted in the plant, and as a result now the plant have shown the pest control for different type of pests. So, this particular area has really kind of transformed, has been very revolutionary, because now large acres of the field where these plants are grown could be protected from these kind of infections, and therefore, your the yield of the food can be very high. However, people have expressed concern that what are the potential risk of using these transgenics in the field, and especially transgenic insecticidal crops. So, one of the potential region could be that this transgene could escape into the wheat population, depending on the outcrossing from the other crops which are in the same neighboring regions. The secondary pest may also proliferate, because if you are controlling one type of pest, probably another type of the pest population might also increase. The cry proteins might also get accumulated in the soil over the period of time. There could be some sort of unanticipated reactions to the human, like which could be allergic reactions, and these insect population could also become resistant to these cry proteins over the period of time. For example, a few cases have been reported where even plants have shown the insects have shown the resistance towards these cry proteins. So, this must be I must admit, this is one of the areas where people have both pros and cons for the technology. There has been large cases where you would have heard that because of the BT crops, lot of farmers committed suicide. Sometimes the big multinational companies, they try to make lot of money out of this proprietary what they have from the licenses. And as a result, on one hand, when we have seen that the huge benefits because of the increased yield which has obtained because of these transgenic crops. On the other hand, there are certain regulatory issues which are still need to be handled. There are certain ethical issues which still has to be addressed. So, this area I will leave that up to you to read more about it, to get more educated in that particular direction, and to find out kind of what you think is the best approach of these kind of transgenic crops. But scientifically speaking, these transgenic the GMO crops have really made lot of increase in the nutritional value, increase in the certain components which can show lot of increased productivity for the plants. How we accept as a human being is something which remains a topic of discussion further. So, in summary, I think in the last several lectures we have been talking about the gene cloning technology, the polymerase chain reaction, different ways of different lab techniques which we have given you demonstration to make the cDNA and do all kind of separation. All of those knowledge moving as a part of the genetic engineering technologies is showing lot of application, whether you talk about the human health or we talk about the plant biotechnology. I am sure you can understand that by just manipulating the genes of interest, by knowing that what we are inserting, where we are inserting, and what kind of trait they may have, I think has made huge and the tremendous changes in overall applications. So, in today's lecture, in summary, we have talked about how genetic engineering technologies could be used to move the genes from one to other organisms, and we have shown you a couple of examples of the salt tolerant plants to the golden rice plants, and also looking at the the crops which are having highly insecticidal resistance properties. The genetic modified plants have definitely lot of potential for increasing the quality and quantity of the food worldwide, and there has been many example of the BT crops. But as I mentioned that how to take these kind of technologies forward, and what are their ethical and economical consequences of that. I think this is an area of discussions, and one need to be very open to look into the pros and cons of these technologies. Thank you very much.